Welcome everyone to the second contributed talk session. We have another 4 wonderful speakers. Please remember to post your questions in the Pathable poll and vote on questions that you want answered. Also, please remember to note in your question which speaker you're addressing the question to. We'll have 4 talks, each 10 minutes, and then a 15-minute Q&A. So our first speaker is Daniel Bunis.
Cool. So hi, my name is Dan Bunis. I'm a grad student for at least the next month. Then I'll be a senior biological data scientist at the University of California, San Francisco. And I'd like to thank the organizers for giving me a chance to be here today to talk about the universal user-friendly single-cell and bulk RNA-seq visualization toolkit that I built and got into Bioconductor in this last cycle called dittoSeq. And I know universal is an adjective that we don't like to throw around typically, but I hope to convince you not only that it is fitting for dittoSeq, but also is a reason to test out the package and hopefully use it for your own RNA-seq analysis. But some reasons that I consider dittoSeq to be universal are its widespread utility. I built a bunch of diverse and powerfully customizable plotters, as well as its broad user space. It is color blindness friendly by default and also robustly documented with new users and novice coders in mind. And then also it has broad pipeline compatibility because I made it to natively handle the most common single cell RNA-seq and also bulk RNA-seq data structures in R. Here I'm showing what I consider to be the main workhorse functions of the package. There's ditto-dimplot, which is for overlaying continuous, like gene expression, or discrete, like groupings data on top of t-SNE, principal components, or UMAP plots. There's also ditto-plot, which is for visualizing continuous variables like gene expression, or say a score you have stored in your metadata, grouped by whatever discrete groupings you want on your x-axis for both the groupings themselves, and also they can be colored by any discrete grouping. Then there's also ditto-bar plot, which is for visualizing discrete variables like say clustering across each of your samples, ages, or any other discrete grouping on the x-axis. But I'll just pause on this one because I think it's a function that a lot of other visualization tools I've seen don't have, but it's really important to check for QC whether you have clusters, say, that might be just within one sample for perhaps just batch effects or some non-biological reason. I also built a ditto scatter plot function. Here I'm showing gene by gene, but you could also imagine showing gene by, say, a pseudotime variable, any continuous variable. And it also works with all the same additional features of ditto-dimplot, like coloring or shaping or fasting. They all work here too. And then more recently, I added hex bin versions of both the scatter plot and the dim plot. Just replace plot with hex, and you can do this. And you can see here how it allows you to not only summarize your data, but also see the density of cells that you have in these distinct regions of the plot. Also ditto heat map, but I also built in a bunch of other functions I'm not going to give examples of. But I think the major draw for ditto heat map is that it works directly with all the subsetting, and it allows you to automatically generate annotation bars if you just give a list of metadata. And I'll say currently it's a wrapper sort of around the heat map package, but I do plan to make it compatible with the complex heat map package in the future, just to give even more customizability to that function. But just to give some flavors of how customizable I tried to make each of these visualizations, and I did that using discrete inputs so that it could be robustly documented for ease of use and learning, but also I built discrete inputs so that you can continue using sort of RNA-seq jargon instead of having to switch to ggplot jargon, which can be quite different. But just to give an example using dittoplot, you can adjust the data representations that are used between violin plots, box plots, ridge plots, or jitters for the individual dots, the individual cells. And here, I guess you can't see it now, but the default was to put the box plot it was covering the violin plots, but I made it so you can adjust the sizes, line weights, colors of each of these representations. So here I made the violin plots wider, the box plots white and see-through, and then by making the jitter dots smaller, it becomes a much prettier picture. But then that wasn't quite enough before I submitted this figure in part of a recent manuscript submission. I also adjusted the titles, subset to specific cells, adjusted the order and labelings of the x-axis groupings, and the y-axis tick marks. And all of these, again, with discrete inputs. Just to give a couple more flavors you can also label and or circle your groups in dim plots, which is especially useful for color blindness friendliness for anyone who maybe won't be able to match the groupings directly to the colors that they go with. But those labels will repel each other in the walls by default, but you can turn both that off and the highlights behind them. And also you can add trajectory inference on top of any dimensionality reduction, as well as many other customizations. And I'll just touch on this one last one by setting data out equals true. For any of the functions, you can access the underlying data, which is both useful for submitting to journals that require that, but also for adding on extra layers like ggplot layers, because they all output pure ggplot objects normally. You can access the underlying data in order to make, say, any alteration you might need before you add your next layer, you can do that. So that's the widespread utility, but I also made it to work for a broad user space, in part by making it color blindness friendly by default. And this is actually, it's part of why I created the package in the first place, because I needed it myself. But the way I did this was by starting with this Okabe Ito color panel, which is equally accessible to individuals with the most common forms of color blindness, as well as to color vision typical individuals. And then I extended it to 40 colors via lighter and darker repeats, which isn't really perfect, but it does allow you to capture the sort of complexities of larger single-cell RNA-seq data sets in still a pretty good way. But I added in other features like increasing the sizes of the legend symbols by default. And this is something that, at least for me, can be helpful if I struggle with the colors. It makes it easier to figure out what's what. Also, color alternatives like letter overlays. You can have A, B, C, D, E, whatever, show up directly on top of the cell's dots. You can shape the cells, use shapes instead of colors, and also faceting. So you can have, say, 5 of your groups on one plot and use the same colors for 5 separate groups on another plot. And then also labels and ellipses, which I already showed. And finally, with a simulate function, I give color typical individuals the ability to assess what their plot might look like to a colorblind person. So these aren't perfect. Like, for example, the color panel isn't quite amenable to people with yellow-green blue-yellow color blindness, sorry, but I do plan on adding additional features as well to try and help with that in the future. But just to touch on what I think is one of the biggest draws as well, the pipeline compatibility. dittoSeq works natively with both single-cell experiment objects and also Seurat objects. And this is important because it reduces the need to convert back and forth between the two, which can sometimes be error-prone. But also for sort of novice users or novice coders who I think we all know often, unfortunately, start with Seurat instead of some of the bioconductor packages that utilize single-cell experiments. I think having a visualization tool that works exactly the same way for both helps people jump back and forth between the two and decreases the activation energy needed to learn an analysis tool that requires the single-cell experiment object. But then also for bulk analysis, dittoSeq powers side-by-side analysis of single-cell and bulk data through use of this import ditto bulk function, which will convert a summarized experiment, DGE list object, or raw counts matrices into a single-cell experiment, allowing dittoSeq to work with that data as well. And this DIFFERENTIal uses a summarized experiment extension. DGEList is the object used by EdgeR, and then lemma, typically users will use raw counts matrices for this. So this gives compatibility with all of the most commonly used differential expression tools for bulk RNA-seq data. So with that, I hope I've convinced you that dittoSeq is pretty universal for all of these reasons. And I'd like to acknowledge my labs. I'm co-mentored by Marina Sirota and Trevor Burt. And I'd like to acknowledge my collaborators, Jared Andrews, Gabriella Fragadiakis, and also acknowledge some generous help and feedback that I've received from Rebecca Yatsik, Giuseppe D'Agostino, Aaron Loon, and Dror Asa. And with that, I'd like to thank you for your attention.
Wonderful. So that was a great start. The next presentation is from Koen van den Berge, and I need to start that video.
Hi, my name is Koen van den Berge. I'm a postdoc at the group of Sandrine Dudois at UC Berkeley. And today I'll be talking about interpretation of single-cell RNA-seq trajectories using differential expression and differential progression analysis. So the advent of single-cell RNA sequencing has really allowed us to study these dynamic changes in gene expression where, for example, stem cells are developing into mature cell types. I will talk about these data sets, and I will mainly focus on one particular data set, which is a data set produced by the NILab at UC Berkeley. So the NILab is studying the mouse olfactory epithelium, and in this specific data set, they have injured the mouse olfactory epithelium in order to activate its regeneration. So this is a 10x data set consisting of about 20,000 cells. And so if you look at this epithelium, you will see here at the bottom right of the epithelium, you have the horizontal basal cells, which can develop into either the sustentacular cells, which are really the support cells of the epithelium, olfactory sensory or olfactory receptor neurons. And then finally, upon activation, upon this injured treatment, they also allow them to regenerate new horizontal basal cells to help in regenerating the epithelium. So if we take a look at this olfactory epithelium upon injury, so here on the right-hand side is a 3D UMAP plot with the slingshot trajectory plotted. So the trajectory starts here in green, which are again the horizontal basal cells, and we have three lineages coming out of the horizontal basal cells. So I will refer to a trajectory as a collection of lineages. The first lineage ends up in the blue cluster, which are the horizontal basal cells, the renewed horizontal basal cells. Then you have this second lineage going into the gray cluster, which are the sustentacular cells. And finally, we have this long stretch here, this long lineage, which are the neuronal cells going into immature neurons in purple and finally mature neurons in orange. So once we were able to identify this trajectory, then the main biological questions of interest was discovering the genes that are responsible in development for each of those cell types. And possibly, can we get at the transcription factors that really drive these differentiation processes and where exactly they are important in development? So if you think about these questions, they basically relate back to differential expression analysis, where we want to compare average gene expression between groups of cells. When we started this project, there were basically two types of differential expression methods out. The first one is a typical group-based DE methods, HR, DSeq2, and LimaVoom, who do a really fantastic job on bulk RNA-seq and even on single-cell RNA-seq data sets, but they really rely on an a priori grouping of cells, which really isn't trivial in this dynamical system. And therefore, it basically excludes them for analysis. Another type of methods like McCall and GP-fates are really bespoke trajectory-based differential expression methods. And they improve upon these group-based differential expression methods because they smooth the gene expression profile over pseudotime, right? The trajectory identifies kind of a pseudotime for each cell, which relates back to how far it is in development of a particular lineage. And then we can smooth gene expression over pseudotime and do differential expression on the smoothers. However, the methods that are out there, they basically only allow us to test for global tests. So they only allow us to test for any differential expression pattern between the lineages or within the lineage, but they don't really tell you where exactly the differential expression is occurring. So in order to allow for more detailed interpretation, we've developed trajectories. So trajectories is a negative binomial count model that smooths the gene expression profiles using that negative binomial distribution over pseudotime for each lineage. So we fit a negative binomial generalized additive model for each gene. And if we look at the linear predictor, then you can see that it basically consists of 3 terms. The last term corresponds to a normalization offset to account for differences in, for example, sequencing depth. The second term allows us to input covariates into the model, patient effects, batch effects. And then the first term is really what does the smoothing of the gene expression profiles over pseudotime. First, before we really smooth these gene expression values, we've got a way to basically assign cells to lineages. And the way we do that is that we use the output of the trajectory inference methods to really assign cells to lineages, because often these trajectories come with kind of weights for each cell to belong to a particular lineage. And then we do the smoothing for each lineage. And the way we do that is that we decompose this smooth expression profile. So we use a basis function expansion of k cubic regression splines, where each cubic regression spline is represented by this BKT here, which each have kind of a parameter that basically defines their contribution to that smooth expression profile. And what's nice about this model is that we can focus the statistical inference on these parameters to allow for detailed interpretation. So under the hood, trajectories is using MGCV, which is a great R package for fitting generalized additive models, which has been developed by Simon Wood. So what that really gets you is, so here on the left-hand side, we've got gene expression of the gene transcription factor. The x-axis is pseudotime for each cell. The y-axis is the log expression, and cells have been colored according to their cell type. Fitting this model is implemented in a fitgam function in trajectories. And so once we fitted the model, we can plot these mean expression smoothers, and you can already see some nice patterns popping up just by estimating these mean expression smoothers over pseudotime. So trajectories is modeler. We allow input from basically any trajectory inference methods. We've got custom workflows for slingshot and McCall that are very easy to use. But we also allow for general input as long as you have a vector or a matrix of pseudotimes. Once we have fitted these models, we've got a range of differential expression tests, which we can basically classify into 2 different classes. And for each test, the plot shows the most significant genes based on this data set. So this is the first step of interpreting these trajectories. The first class is within lineage comparison. So comparing gene expression within, for example, the neuronal lineage. The first test is a start versus end test, which basically allows you to compare any 2 pseudotime values or even regions of pseudotime values within a lineage. By default, it compares the start and the end of the lineages, which is why we're calling it the start versus end test. The association test is a generalization of that and really checks whether mean expression is associated with pseudotime. So is the smoother significantly varying over pseudotime? The second class of tests are between lineage comparisons. We start off with the diff-end test, which basically compares the expression profile at the ends of these lineages, so often at these mature cell type stages between the different lineages. And then the right-hand side is again a generalization of that, the pattern test, which really allows you to test for any differential expression pattern over pseudotime. We can focus that test again on a particular pseudotime region, and that's the early DE test. So other things we can use these smoothers for is, so the second question of the biologists was, can we get at these transcription factors that may be involved in the development of each of those lineages? So what we did here is that for each transcription factor, we use these smooth expression profiles we've estimated using trajectories, and we check whether it's significantly peaking somewhere along development. If it is, then we retain it and we check where exactly the most significant peak of that transcription factor is occurring. And here on the left panel, I'm plotting these standardized mean expression profiles of these transcription factors we've identified. On the x-axis is again the pseudotime, and these expression profiles have been colored according to where we identify their most significant peak. So you can see the blue transcription factors peaking first, then the yellow, and then the red. The right-hand side is another visualization of this, where we order the transcription factors according to their expression peak. So just to quickly finish off, what we're currently working on is basically trajectory inference and differential expression across conditions, where we may have multiple data sets with a similar trajectory, and we check whether we can fit one trajectory that includes both or all of the conditions. And if we're able to do that, then we can answer questions like differential progression, which is really getting at whether the cell density along pseudotime is similar for both conditions, meaning whether cells develop in similar ways along the trajectory or not. And then also differential expression, for which we have basically extended trajectories to allow for a condition effect within each lineage that allows you to estimate a condition-specific smoother for within each lineage. And then we can again use these differential expression tests of trajectories to interpret these trajectories. So if you want to know more about that, please do check out our workshop on Friday. So finally, I'd like to really thank the DIFFERENTIAL team, the Nile Lab for generation and interpretation of these data sets, and the DIMVERS team who helped us out for simulations. So DIFFERENTIAL is up at Bioconductor, and you can also read the paper if you like. So thanks for your interest, and I'm happy to take questions.
All right, wonderful. Another great talk. Our third speaker is Will Towns.
My name is Will Towns. I'm from the computer science department at Princeton. I'm going to talk about two techniques for scaling methods for really large data sets, just using dimension reduction as a motivating example. So first of all, a lot of the tasks that we always talk about in bioinformatics and genomics really boils down to solving some optimization problem, whether it be clustering, dimension reduction, or differential expression, which is basically a regression problem. The big idea is we want to define an objective function or a loss function that describes how well a particular unknown parameter describes the data. So this function is going to be taking as input the data and some initial guess as the unknown value of the unknown parameter, vector, or matrix, or whatever. And then it's going to, iteratively change the unknown parameters and try to either maximize or minimize the loss. So as an example, you can think about a Poisson generalized linear model, which is very simple, but hopefully it will be good for getting the point across with some simpler math equations. If we have n observations of some data, x and y, we want to figure out the best possible values for the regression coefficients. We can define the outcome as being a Poisson draw from some mean mu I, and the mu I is connected to the covariates and the parameter through this link function. So then this is just the log likelihood of the Poisson up to a constant, of course. You can ignore additive constants. And we want to maximize this log likelihood. So the way to do it is for differentiable functions, you can just take the derivative, and then that tells you which direction you want to go to try to climb the hill or go to the bottom of the hill, depending on which direction you're trying to solve. So this is the closed form derivative for the Poisson GLM. It's very simple. And the key thing to note here is for each iteration of our optimization routine, we have to actually compute over the entire data. So we're using all of X and all of Y to get our gradient. And so this is really where the problem occurs for big data, because potentially we're going to have to do many iterations of gradient computation and updating this mu(i) each time. And so that could be quite slow. If every time we evaluate the gradient, we have to compute over the entire data set. And it might also be difficult because the data may not fit in memory. So the main idea that I'm proposing, which is a very old idea, I didn't come up with it, is basically we're going to work with small subsets of the observations instead of the entire data set. So this is often called mini-batch. We can basically split all the indexes into non-overlapping sets, and each one of those sets of indices is a mini-batch. I'm just using sequential splitting here, but in practice you would usually do a random split. So the first of the two methods that I want to describe, and these are fairly general, I'm trying to get the concepts across so that other people could maybe use them in their algorithms. But just to illustrate it, I'll stick to the Poisson GLM example. So very simple, we can take this sum over all observations and split it up into an inner sum and an outer sum. So this is exactly the same thing, but we're just changing the ordering. So here we're going to sum over only the observations that are in a particular mini batch, and then to get the total, we sum over the whole mini batch. And so if we treat this inner sum as a sufficient statistic, or SB for sufficient statistic, that inner sum is actually pretty cheap to compute because we only need a very small subset, maybe 100 or 1,000 observations instead of 1,000,000, which saves us a lot of time. So basically at each iteration, we only update one of these SBs and hold the other ones constant and compute the gradient with that modified total gradient. So this trick was first proposed, from what I can tell, by Michael Hughes in the Bayesian nonparametrics literature. So you can check out, he has 2 papers using this in Dirichlet processes, but it works with any GLM type exponential family likelihood. So another approach, which is probably better known, is to, again, here's the true gradient. We're just going to sum over the mini batch, and instead of caching the sufficient statistics across mini batches, we actually just pretend like this is the full data set and multiply it by a scaling factor. So this in expectation is the same as this, but it's not actually the same. So it's sort of a unbiased estimator of the gradient, which converges as the size of the mini batch increases. So the rationale behind that is just simply the law of large numbers. And this technique is very commonly used in deep learning. It works with any differentiable loss functions. It's more general than the memoized approach. But there's some issues of the noisiness of the gradient estimator. So here's just a summary of some of the pros and cons. From a theoretical standpoint, because the stochastic gradient doesn't need to do any caching, it actually has a more favorable memory growth profile, and it's easier to implement. But on the other hand, it's a lot harder to tell when it's converged. So here's an example of the trace plot of the objective function value for a Poisson GLM PCA, which is a generalization of PCA for non-normally distributed data. So the thing you want to minimize is the deviance, which is basically the negative log likelihood. You can see the trace plot of the objective function for the stochastic gradient approach is extremely noisy, and it even actually increases a little bit sometimes, and it bounces around. It's kind of hard to tell when it's leveled off. In contrast, the full gradient approach, the blue, which is labeled none, as well as the memoized approach, they both decrease very smoothly, and it's quite easy to tell when they've converged. So I tried these different implementations with the 10x PBMC data package, and you have data starting from about 3,000 cells that goes up to about 68,000 cells. And as you can see, the memory consumption of the full gradient optimizer increases quite rapidly. Whereas if you use either the memoized or the stochastic gradient approach, it grows much more slowly. I was surprised not to see the quadratic increase for memoization. Don't really have an explanation for that, but it seems that, you know, from a very rough sense, either one of those approaches would probably be fine. I don't know, so each panel is a different mini batch size. I don't really have an explanation for what's going on in this middle one, but yeah, future question. I also profiled the speed, how long it takes for these different algorithms to converge. And here the stochastic gradient approach was a clear winner, but I cheated by running the memoized approach first. And once it converged, I just set that threshold. So as soon as the stochastic gradient reached that threshold, I terminated it. I didn't actually try to figure out whether it converged or not. So if you were trying to assess convergence, this thing might run a lot longer because it's hard to tell when it's converged. Yeah, and the speed of the full gradient approach was pretty comparable. So if you want to try this out on your own data, both memoization and stochastic gradient have been implemented in the GLM PCA CRAN R package. Stochastic gradient is also used by the Minibatch k-means, and their paper, that's Davide Riso and Stephanie Hicks. I'm sorry, I don't remember the first author, but you can look it up on Bioconductor. But that paper has a lot more extensive memory profiling and looking at things like HDF5 files and you know, the chunk geometry, which is also very interesting. So thanks for your attention, and I'll be happy to take questions later on.
Wonderful. Thank you for a very interesting talk. Our final presenter in this session is Lauren Su.
Okay, hi, my name is Lauren Shu. I am a graduate student at Harvard School of Public Health, and I work with Aideen Colhan at Dana-Farber. And today I'll be presenting on Corral, which is a new bioconductor package that performs matrix factorization methods for dimension reduction and alignment of single cell omics count data. So as we've been talking about in the session, single cell data are really rich in information, but pose these unique challenges, especially in their large dimensionality. So it's especially important to be able to perform effective dimension reduction of these data. And when we look at a lot of pipelines, we can see methods like really old and basic methods like PCA being used a lot, and with little consideration of whether these approaches are the most appropriate way to reduce dimension in these types of data. So in looking at PCA, one popular way to compute it is, there are a lot of ways, but one is to use SVD. And in that approach, the matrix is z-score transformed and then decomposed into 3 matrices comprising the left and right singular vectors and the singular values, as shown in this cartoon. We in our recent mini review, we were really interested in looking at the impact of preprocessing steps on results. So this is a figure from that mini review, and we took a benchmarking data set from the CellBench data package, which comprises 3 lung cancer cell lines sequenced on 3 platforms. And we first performed SVD on the raw counts and the log counts, and then also performed PCA to see what is the impact of this z-score transformation. And as we can see, when we perform PCA on the log counts, then we can get a reasonable clustering of the cells, as well as some alignment across batches. However, when performing SVD alone on these data, it really highlights the importance of the centering step in particular, as we can see that in the first and second components are somewhat correlated. So in thinking about this, we wondered if there's a perhaps more appropriate transformation to perform to the data prior to performing SVD. And correspondence analysis is an old method. It was developed first in the 30s and was repopularized in the 50s and 60s by the French School of Multivariate Statistics. And it's a matrix factorization method designed for sparse count data. It's related to PCA, as you can see here. It's one of the algorithms for computing it is really similar, but rather than performing a z-score transformation, we perform a chi-square transformation prior to decomposition with SVD. And it's been applied in a lot of other fields, including ecology, archaeology, linguistics, and to a limited extent within bioinformatics previously, especially for bulk data. So there are a few packages in R that already implement this method. However, none of them are designed for use on data that is the size of a single cell matrix. So we developed the corral package that uses sparse matrices, applies fast SVD approximation. We currently use the ERLDA implementation. However, it's modular, so we can substitute any other methods that emerge or anything like that, and interacts directly with bioconductor objects, including single cell experiments, summarized experiments, and multi-assay experiments. It's currently in the devel branch of bioconductor. So within this package, we have two main methods. The first is called corral, which implements kind of this vanilla correspondence analysis, but is designed to perform it on much larger data sets than is typically analyzed in other fields. And the second method is called corral M, which we developed as an adaptation of correspondence analysis for alignment of multiple matched tables into a shared latent space. So I'm going to first walk through the math for corral, which is just correspondence analysis, and then I will walk through corral M as well. So as I mentioned before, the first step is a transformation of the counts from count matrix into standardized residuals. And so basically this involves first computing this pi ij for each value, which is the abundance. And then from that abundance table, we compute the row weights and the column weights. And these are used to compute that expected value. And then we do observed minus expected over expected to find the chi-square statistic as shown here. And then we take this matrix of the standardized residuals and perform SVD on that. And then that provides the embeddings and the feature and cell embeddings. And the usage of this function is very simple. It's just using corral, and it can be called on a matrix-like object or single cell experiments or summarized experiments. So the corral M method performs alignment and integration and begins with a really similar preprocessing step. However, one notable difference is that whereas correspondence analysis scales each table necessarily using its own row weights, there's also an option to kind of force the integration more by setting the contribution of each table's row weights to an overall shared row weights that is used to compute the expected value of the statistic for this formula. And similar to the other method, there's only one more step. We then, after computing these tables, we then concatenate the transform matrices matching on, in this case, as I'm describing, features. However, it could also be matched on cells if we're talking about multimodal data, but if we're focusing on matching on features. And we then decompose that. And the usage for this method is similar. And again, it can be called on a variety of types of objects, including lists of matrices or single cell experiments, multi-assay experiment, and also a single cell experiment when provided with which attribute from call data is the batch indicator. So I'm going to present some results in a few benchmarking data sets. First is the ZengMix data set from the duo clustering package to just very briefly show correspondence analysis. And then second and third, I will be talking about the corral M results in the SE mixology data set from Cellbench and the PBMC data set from Seurat data. So the ZEGMIX dataset comprises 8 pre-sorted cells, different immune cells that were sequenced on 10x, and then recombined into 3 datasets. So ZEGMIX 4 EQ has them in approximately equal proportions, but for the sake of time, we're going to focus on just the 4 NEQ, which looks at 4 cell types in unbalanced proportions, and 8 EQ, which looks at 8 cell types in all 8 of the cell types in approximately equal proportion. So in comparison against PCA, we can see that the correspondence analysis approach is robust to not performing a log transformation, which, as we heard in the first keynote, is potentially not appropriate for count data. And we can see there is, whereas if we look at the results from PCA, we can see that the output really does require that log transformation in order to separate the clusters. So this is just really presenting an existing method. What we're really excited about showing is the integration approach. So to start, I'm going to show the results in the SC mixology dataset, which, as I mentioned before, comprises 3 lung cancer cell lines on 3 technologies and is a total of around 1,400 cells. So these are the results from the mini review paper I mentioned before, where we were really looking at PCA and SVD. And if we compare the corral M approach against the results from using PCA, we can see that we have a much better integration across the different batches. And again, it's robust for use on both counts and log counts. However, PCA is not really designed to do integration, so we were also interested in identifying a couple of comparison methods that were actually for integration. So from a benchmarking study from Tran et al. Earlier this year compared 14 integration methods and found that Harmony and Seurat are 2 of the 3 recommended methods. Briefly, Harmony performs PCA, then uses the embeddings from PCA to perform iterative self-clustering and actually can accept any embeddings that a user might provide. And then the Seurat pipeline includes SC transform, which does a normalization variance stabilization and feature selection based on UMI matrices, and then performs integration using canonical correspondence analysis, CCA, and with an anchoring scheme. So when we compare the results from these different approaches, here are the results from corral on the top. Again, that we saw before. We can then look down and see the results from Harmony and notice that the method is not able to integrate these data. However, when we perform corral with Harmony, then we can see that it actually helps to separate the clusters better and improves both sets of results. Just looking at the plots themselves, Seurat has the best results. However, if we look at the computation time required to achieve those, it is many-fold, the corral and Harmony, and even performing both of them together. So in the SC mix data set with only around 1,000 cells, it's not a big deal. But if we are looking at anything larger, like for example, this PBMC data set from the Seurat data package, which comprises 30,000 cells of 9 cell types in 9 batches from 6 technologies, that computation time will scale not favorably. So to look at the results here, first we can observe that Harmony is not successful. Harmony as written is not successful in integrating the batches here, nor in identifying clusters. Contrary, we can see that Seurat is able to identify the different cell groups. So comparing the Seurat results with corral, we can see that they may look pretty different at first, but if we kind of go through them by the different cell types, we'll see that they are fairly similar. So starting with the blue B cells, we can see that both approaches are successfully separating this group. We can also look at the T cells and NK cells and see that we have a similar geometry in UMAP space, resulting from both methods. And finally, looking at this kind of larger complex of the monocytes and the dendritic cells and the megakaryocytes, we see that each of these are similarly grouped out. However, looking at just these embeddings can be deceptive since there are so many cells. So in order to verify that we are actually integrating, we also take a facet view to see where the individual cells lay within each batch. So in comparing these results, you might say, okay, what is the big difference? Well, the really big difference is the computation time required, as I mentioned. Corral actually is able to create this embedding in about 5 seconds, whereas in order to create the low dimensional representation for the embedding on the right, Seurat actually takes around 30 minutes. So it's kind of illustrated heuristically with the cartoons here. And in summary, correspondence analysis is a matrix factorization method for count data, and we suggest it may be more suitable than PCA for scRNA-seq data. The package corral integrates with bioconductor, is fast, scalable, modular, and also includes this set of corral M approaches. That's still an active area of development for us. And yeah, so to download the package, here are the commands. And then if you're interested in talking more about PCA, matrix factorization and correspondence analysis, I encourage you to attend our session on tomorrow at 1 p.m. Thank you.
Wonderful. Thank you for a great talk. And actually thank all 4 speakers for a very interesting session. We have time for some questions. Keep them coming in through the Pathable poll, and we'll try to get to as many as we can. So the first question for Dan, do you have plans to extend dittoSeq to spatial data like Visium and Nearfish?
So I would say yes. I think if you can transfer the X and Y or Z, like the Visium coordinates into metadata, you can actually work with them already. I haven't worked with that data, so I don't know the structures that are used, but if it's something that is easy to translate, I would definitely be interested in making that even easier.
Wonderful. So the next question is for Koen. Do the statistical tests account for the fact that cells are not independent replicants?
That's a really good question. And I think there's multiple levels to the question. So first of all, because you do the trajectory inference, you're kind of inducing some kind of correlation between the cells, as in, you fit the trajectory in a reduced space, and cells with a similar pseudotime are expected to have similar gene expression profiles, and this is actually what makes the smoothing useful. I'm assuming that the question is rather pointing at, say you have 5 patients and you sample cells from each of the 5 patients, then the cells can be considered pseudo replicates for each patient. And no, we do not account for that. We currently do not have a way to account for that. We allow you to add the patients as fixed effects, but that can only take you so far. But if you're interested in analyzing these multi-patient data sets, I recommend the Muscat paper and package from Helena Crowell and Mark Robinson's lab. And I should say that we're definitely interested in allowing to account for these effects.
All right. So the next question is for Will. I'm actually going to combine the top 2 questions because I think they're related. So the first one is asking about how you define the mini batches. And the second one asks, working with mini batch, do you select them randomly? If you have a data set enriched in a specific cell type, for example, brain where 90% plus of cells are neurons, how do you ensure that you capture all of the variants?
So could you clarify I don't really understand the first question.
So I think both these questions are getting at how do you select the mini batch?
Sure. So first of all, I just really want to promote the mini batch k-means paper, which did a much more thorough job comparing the pros and cons of smaller versus larger mini batch sizes and different schemes for choosing the mini batches. Than what I did in my cursory analysis. But so if you're worried about missing out on the rare cell types, you can guarantee that every cell gets represented once per pass through the full data set by forming this partition, right? So especially with the memoization approach, because there you really are going to include every cell equally. You're just sort of amortizing the inference over more iterations. But in terms of the stochastic gradient approach, you can do something like establish a schedule where in the first full pass through the data set, you shuffle all the labels and then split equally into chunks. And then you just iterate through those mini batches. And then once you've gotten through the whole data set, then you do another round of shuffling and splitting. Or another approach, so that approach would guarantee that every cell gets its fair share. But that's also requires like a little bit slightly more work. So the fastest and easiest way is you just literally draw a random sample of the cells uniformly at each iteration. But that does run the risk that there may be some cells that actually never appear in the mini batch. So there's certainly many different ways to do it. I tend to prefer the partitioning and shuffling approach. But I would encourage folks who want to try it, definitely experiment and try it on very simple toy data sets to make sure it's doing what you think it's doing.
All right, so the next question is for Lauren. Corral looks fast. How will it scale to larger data sets? Have you tried it on 1 million neurons? Are there ways to make it faster?
Yes, so actually one of the things that we're excited to work on next for Corral M is to think about how we can use matrix projection methods to use something similar to the kind of memoization approach that Will described to further speed up the process. As it is now, we're currently using ERLBA for decomposition, which does not scale terribly well to 1 million cells. So that is definitely an area of interest for further development.
Great. So the next question is actually for two of our speakers. Can we integrate TradeSeq with dittoSeq and see which genes are differentially expressed along the trajectories? I don't know, Dan, do you want to take the first crack at this?
Yeah, sure. So I think perhaps the first step of this would just be sort of using the pseudotime that you get out of TradeSeq. You can integrate that into the scatterplot function as sort of the x-axis, and then I would say that you can sort of take advantage of the trajectories that I have built in already to add the vectors and display lines on top. I'm not sure, we could potentially work together and make that even more robust, but yeah, that's what I would suggest for right now. I don't know if you have more to add, Koen.
Yeah, I'd say, you know, it's definitely worth combining. You know, you can do the statistical analysis with TradeSeq and then look at your results using dittoSeq. Yeah.
Great. Let's see. So, Koen, can TradeSeq be employed in multi-condition, multi-replicate case control single cell designs, like healthy versus vehicle versus drug treatment dosing with high sparsity?
That's a complex design to begin with. So the multi-condition case, we're working on it as I mentioned in the last 2 slides, and actually we have a workshop on that on Friday. So if you're interested in how we, you know, can potentially tackle that, I recommend you take a look at the workshop. So we've got something put together for that, and you know, the multi-replicate scenario again seems to refer back to the first question, so I'll refer to my answer there, where you know you can add fixed effects but no random effects, for example. Yes.
So Will, the next question is wondering whether there's an article or an online resource elaborating on the approaches you described.
There's a smattering. So first and foremost, the stochastic gradient is just ubiquitous in deep learning. So anytime you want to use TensorFlow or PyTorch or any of those types of things, you're going to have to tell it what kind of optimizer to use. And it'll have a flag in the Python function or the R function that says, how big do you want your mini batches to be? So you can start by, for stochastic gradient, you could start by just looking into the documentation of some of those standard deep learning libraries. For memoization, I just came across it through the Bayesian nonparametrics literature, and it was quite obscure. But if you just look for Michael Hughes, he's the one, as far as I can tell, who came up with this idea. I think it's a really cool idea, but I have not seen it used really anywhere except for in his 2 papers. So I kind of wanted to just promote that idea because I think it's much more general than just for Bayesian nonparametrics. You could use it for linear regression, you could use it for PCA. Although based on those benchmarkings, I'm not sure that I'm going to use it because it actually seemed to be quite slower than stochastic gradient. Yeah, unfortunately, I can't point to an authoritative reference, sorry.
All right, so maybe one final quick question, and then I'll just plug that all of these questions are saved. You can reach out to the speakers on Slack or through Pathable. We do encourage you to follow up if we didn't get to your question. So Lauren, have you compared Corral-M to neural network-based data integration methods like SCVI?
I have not specifically compared it to SCVI, but that is a great suggestion, and we'll definitely look into that.
Wonderful. Thanks for being quick and concise to finish us up. And thank you to all 4 speakers again for a wonderful session.